Breaking the wall of the greenhouse microcosm. Derek Lundberg, Max Planck Institute for Developmental Biology, Tübingen.
Okay, so thanks for inviting me. 70% of the world's fresh water is currently used for agriculture, and the world's population is increasing, and we're basically out of farmland. So there's a tremendous need to improve the efficiency of the crops that we have. And this really falls to genetics. We've done this since ancient times by breeding the best plants for the next generation. But this is slow and we didn't really know why it worked. What geneticists have done is make pets of some organisms, such as the fruit fly or the Arabidopsis thaliana, a small plant, or corn, and bring it into the greenhouse, which I'm using as a metaphor for a structured lab environment. And then we've studied these things in great detail in the lab, sequenced their genome, invested these resources, and then we understand how they work in pseudo-nature. And I say pseudo-nature because a greenhouse doesn't have, or at least a good greenhouse doesn't have deer, squirrels, snails, wind, rain, and all the microbes that are out in nature. And so what happens then when we want to see if our discoveries matter outside is a lot of times they don't because of these things that we were neglecting. So my solution is to break greenhouses, not literally. I'm just saying, like, not all of them either, but I'm saying that to actually do the experiment that nature designed as much as possible. And really this is all a function of modern DNA sequencing because all the resources that we had to invest in the model organisms that we studied for these genetic discoveries, now you can generate quite quickly because the computational power and sequencing power is there. So my project, briefly, is I'm going to a field where there's lots of plants of the same species, taking those plants, sequencing the plant DNA as well as all the microbial DNA in detail because the pathogenesis can be due to a single gene in a bacteria. And then this is a huge data set, and we're searching this for patterns, such as plant genes that are not associated with pathogen genes or microbial genes that are associated with bigger plants. And these genes are breeding targets or targets for genetically modified organisms in places where these are allowed. So that's another wall that I think needs to fall, but that's not this topic. So anyway, this will explain a lot of mysteries that currently exist about plant immunity and also give us gene targets for agriculture. So, questions?
How would you get the deer into your picture?
The deer, you just do the experiment in a field where there are deer.
The practical use, how do you transport your knowledge into praxis?
So when I find a pattern in the data, I test that hypothesis about that gene by knocking that gene out of that organism and then testing it in one of these more structured lab environments. And then that gene is usable.
Good.